So now we will see the incompatibility of RH factor. When incompatibility occurs is when the RH negative person encounters with the RH positive blood, then incompatibility occurs. So here this incompatibility of RH factor leads to erythroblastosis foetalis. Erythroblastosis foetalis is a hemolytic anemia which affects the fetus or neonate, that is the newborn baby or the baby which is inside the womb of the mother. This is caused by transplacental transmission of maternal antibodies to fetal RBCs. So why it is caused is because of the transplacental transmission. Transplacental transmission means anything which is transmitted from the mother's body to the fetus through the placenta, that is called transplacental transmission. So what is transmitted here is maternal antibodies, that is antibodies of mother is transmitted to the fetal RBC, that is to the RBC of the fetus, and it destroys or causes the destruction of RBCs. So erythroblastosis foetalis is a condition where it leads to certain destructive effects in the child's health. So hemolytic anemia means destruction of RBC or breaking down or lysis of RBC occurs. That is why it is called as hemolytic anemia. So when the RBC level goes down, we call it as anemia. So the child or the baby suffers from a condition called as erythroblastosis foetalis when the incompatibility of RH factor occurs. The disorder usually results from incompatibility between maternal and fetal blood groups of an RH antigen. That is one. So that is when the mother is RH negative and the fetus is RH positive, or vice versa, this condition may occur. Now we will see an example for this. So the mother is RH negative and the fetus is RH positive. So in this case what happens is RBCs from the fetus can go into the mother's bloodstream through the placenta. So here you can see the RBC from the fetus. The fetus blood contains the RBCs. These RBCs, they move into the mother's bloodstream through the placenta. So as a result what happens, the RH negative mother's immune system treats the RH positive fetal cell as a foreign substance and makes antibodies against them. So mother's body will start reacting against the RBCs because the RBC contains the RH antigen. So this antigen is new to mother's body. So the mother's body recognizes it as a new cell or new substance. So it starts working or giving response against that. The mother's body gets sensitized. So it starts producing antibodies against it. These anti-RH antibodies may cross the placenta into the fetus where they destroy the fetus circulation red blood cells. These antibodies which are produced by the mother's body will in turn move through the placenta towards the fetus, and then it starts destroying the red blood cells which are present in the fetus circulation. This condition is called as hemolytic anemia or erythroblastosis foetalis. Now you can see in this diagram how RH hemolytic disease develops. So the father is RH positive and the mother is RH negative. So there is a possibility of the child to be RH positive. So in this case, when the mother is RH negative and when the child is RH positive, this mother's body becomes sensitized because of these RBC cells which are moving towards the mother's bloodstream through the placenta. So RH negative mother with RH positive baby. RH positive baby's blood cells enter mother's bloodstream. So invading RH positive blood cells cause the production of RH antibodies. Now mother's body will start producing antibodies against the RBC of the fetus. RH antibodies remain in mother's bloodstream. Now after that the baby is born, but this first baby will not be affected. Why? Because it needs time for the mother's body to prepare the antibodies. By the time the baby will be born. So first baby will not be affected with this condition. But later pregnancy with RH positive baby. So again when the same lady gets pregnant and when the baby is again RH positive, this mother's body is ready with the antibodies, that is RH antibodies, that will attack the RH positive baby's blood cells, causing RH disease. So, this leads to agglutination of the blood, that is it leads to various symptoms which you will see in the next slide. Now, we will see the effects of erythroblastosis foetalis. So, what occurs is as a result of that, anemic jaundice baby, so yellowing of the body and anemic condition because RBCs are destroyed, or otherwise yellowing of eyes, yellowing of skin and excess bilirubin in blood. Bilirubin is the pigment from the liver. So, that excess bilirubin will be sent to the blood, and because of that what happens is when the excess bilirubin is present from bloodstream, it moves to the brain tissue and it may lead to mental retardation also. So, this condition occurs because of erythroblastosis foetalis. So, what can be done is some exams and tests are there to determine this condition. A positive Coombs test result. So, there is direct and indirect Coombs test. So, these tests are to identify the antibodies present in the baby. Fetal blood sampling for RH sensitization during pregnancy. So, fetal blood sampling, that is testing the blood sample of the fetus. And then a high level of bilirubin in the baby's cord blood. So, when there is a high level of bilirubin, that can also be detected with certain tests. So, what are the treatment that can be given is, the affected baby should be treated with aggressive hydration, that is intravenous hydration will be given. Then early phototherapy. Phototherapy means treatment with light, fluorescent light, that is early phototherapy. So, here if you see, exposing the baby to fluorescent light bulbs or other sources of light, sometimes sunlight also. Next one, yearly exchange transfusion if required, that is blood transfusion if required, that is to remove the bilirubin, to remove the antibodies, that is exchange transfusion helps in removing the bilirubin antibodies or sensitized cells which are liable to be hemolyzed. Hemolyzed means which can be broken down, or the replaces the RBC numbers, that is hemolyzed RBCs can be replaced with the help of the treatment. How can we prevent these conditions is, when the mother is RH negative and the fetus is RH positive, anti-D antibodies are administered to the mother at the 28th and 34th week of gestation. So, that is anti-D antibodies called Rogam will be given. So, if this mother delivers RH positive child, that is the first pregnancy, immediately I told you when the first child is born, that will not be affected, only the second child will be affected. So, immediately when the RH positive child is born, this mother's body will be sensitized. So, immediately anti-D antibodies will be administered to the mother soon after delivery. Why? Because the next child should not be affected. This prevents the formation of anti-D antibodies in the mother's blood by destroying the RH fetal RBC. So, now the fetal RBC will be present in the mother's body. So, that mother's body gets sensitized. So, mother's body will start producing the anti-D antibodies. So, before that we are giving passive immunity by giving anti-D antibodies, so that immediately this anti-D antibodies will destroy the RH fetal RBC, so that mother's body will not start producing this anti-D antibodies. So, this prevents the formation of anti-D antibodies in the mother's blood by destroying the RH fetal RBC before the mother's immune system is sensitized. These are the preventive measures. Next, genetic control of RH factor. So, we have seen this RH factor. Which controls this RH factor? Which are the genes that are responsible for controlling this RH factor? So, two theories are there, or two hypotheses are there. One is Fisher-Race hypothesis, another one is Wiener hypothesis. So, first we will see about Fisher-Race theory of RH inheritance. So, according to Fisher and Race, this RH inheritance is controlled by three closely linked loci. This loci can be pronounced as loci also. Three closely linked loci on each chromosome of a homologous pair. So, here you can see in this diagram. So, this is a chromosome. So, this blue color denotes the chromosome. So, this chromosome contains three closely linked alleles. So, here this three closely linked loci on each chromosome of a homologous pair. Each locus has its own set of alleles, which are capital D, small d, capital C, small c and capital E, small e. So, either of this can be present. So, this homologous pair, if you see, capital C capital C or capital C small c can be present. So it can be present in any condition. So each locus has its own set of alleles. So the D gene is dominant to the small d gene, but capital C small c and capital E small e are codominant, like how we have seen in the blood group. So here which is dominant is capital D is dominant over the small d, whereas small c capital C and small e capital E are codominant. Then the three loci are so closely linked that crossing over does not occur, and the three genes on one chromosome are always inherited together. So they are present so closely, even after crossing over also this cannot be separated, they are as such inherited together. So crossing over, when crossing over occurs there will be change in the genes, but they are so closely packed. So even after crossing over also there will not be any difference, there will not be any segregation, they will be as such inherited together. So you can see when this RH positive, that is when the status is negative and positive, you can see here this gene CDE. So you can see both are recessive D, small d. So the person will be RH negative. If the gene, that is the allele small d is present, both are recessive alleles, then it is negative. The remaining condition you can see either one will be capital D or capital D, both are capital D. So if this condition prevails, the person will be RH positive. So at least one capital D is there, the person will be RH positive. When both are recessive, that is double recessive, R is present, it is negative. So this is according to Fisher and Ray's theory. Next one is Wiener hypothesis. So Wiener, according to Wiener, existence of 8 alleles at a single RH locus. So there they said 3 loci, but Wiener said 8 alleles are present at a single locus. So dominant or produce RH positive phenotype, whereas double recessive genotype, that is smaller, that will denote the RH negative phenotype. So according to Wiener, 8 alleles are there and all these are present in the same locus. There 3 locus, here same locus. So which will determine the RH positive phenotype is capital R, whereas double recessive smaller will indicate the negative phenotype. So this is the genetic control of RH factor. These are the 8 alleles represented by Wiener. So we have seen about the ABO blood group types, then RH factor and incompatibility of RH factor, and then genetic control of RH factor. Next is sex determination. So what is sex determination? The method by which the distinction between male and female is established in a species, that is sex determination. So how we determine or distinguish a male and a female, that is what is called a sex determination. The sex chromosome determines the sex of the individual. So which differentiates a male and a female is the sex chromosomes. Sex chromosomes may be similar or dissimilar. If it is similar, we call that individual as homomorphic individuals and the gametes are homogametic. If it is dissimilar, we call them as heteromorphic individuals and the gametes are heterogametic. So sex determination is important to distinguish a male and a female. So chromosomal basis of sex determination. In that first we are going to see about heterogametic sex determination. Heterogametic means I said 2 types of gametes will be present. Here in that one of the sexes produce similar gametes and the other sex produce dissimilar gametes. So similar gametes means, like if you take a human being as an example, XX in the female, whereas in the male XY. So one of the sex produce similar gametes and the other sex produce dissimilar gametes, and the sex of the offspring is determined at the time of fertilization. So this is heterogametic sex determination. Heterogametic males, heterogametic sex, that is digametic sex, refers to the sex of a species in which the sex chromosomes are not the same. So in male, if you see in human beings, they are not same. One is X and the other one is Y. So X and Y sex chromosome. This would be referred to as the heterogametic sex, and females having 2 X sex chromosomes, that will be referred to as homogametic sex. So heterogametic means 2 different chromosomes. Heterogametic females, if you see, so far we have seen only the males have the heterogametic condition, that is X and Y type, but in birds, moths and some fish, if you see, the females are heterogametic, that is they have two different types of chromosomes, while males are homogametic, that is they produce the same type of gametes, whereas the females produce two different gametes. So they are represented as ZW for females and ZZ for males, like how we represent in human beings XY for male and XX for female. But here if you see, heterogametic condition is seen only in the female. So here they have given ZW for females and ZZ for males, and the mechanism as ZZZW system. So we call XXXY type in human beings, like that ZZZW system here. So in this itself there are two types in this ZZZW system. One is ZZ, Z0, ZO type and ZZ ZW type. So this is XXXO type where the Y is absent, and here ZW type, that is XXXW, XXXY type. So two conditions are there in the heterogametic females. Sex determination in human beings, this already you have learned. So human beings have 23 pairs of chromosomes, in that 22 are autosomes and one is a sex chromosome. So however a pair of X chromosome is present in female and XY in male. So XX in female and XY in male. So during spermatogenesis what happens is the male produce two types of gametes. In that gametes 50% will have X chromosome and 50% of the gametes will have Y chromosome. Or if you see in the female, on the other hand, the female produces only one kind of gamete, that is only one type of ovum, all carrying only X chromosomes. So when the sperm having Y chromosome fertilizes the ovum, the sex of the baby is male, and when the sperm carrying X chromosome fertilizes the egg, the sex of the baby is female. So you can see in this diagram. So male is heterogametic, having two different types of chromosomes. So 44A, A means body, that is carrying autosomes, body chromosomes, and this is the sex chromosome XY. Here in female is homogametic, that is two or same chromosomes, 44 autosomes plus 2 allosomes, that is XX. Sperms, if you see, during meiosis what happens? It will be reduced to half. So 50% will have 22A plus X and 50% 22A plus Y. Whereas here in ova, all that is 22A plus X, again 22A plus X, because they produce only the same type. So now if you see, when they combine together, this and this, when it combines, when they combine together, what will be the offspring's progeny? So 44A plus XX, when this X and X together, when they combine, the female child will be born. And same here also. So 44A plus XY, when it is combining together, male child will be born. So this is how the progeny is established in the human beings. It's about sex determination in human beings. So next we will see Y chromosome and male development. Usually Y chromosome has less functional genes when compared to X chromosomes. So the number of genes in Y chromosomes are very less when compared to X chromosome, because X chromosome has nearly 1000 genes, whereas Y chromosomes, if you see, only 60 functional genes are present. So see, current analysis, it has revealed that numerous genes and regions are present in Y chromosomes. So you can see in this diagram, the first region is PAR. PAR is pseudo autosomal region. This pseudo autosomal region is the place where synapses and recombination occur during meiosis, and this region is similar to the region which is present in X chromosome. So synapsis occurs only in this region, pseudo autosomal region, and the remaining 95% of Y chromosome is referred to as non-combining region, or NRY. So NRY means non-combining region, and in this NRY, if you see, there are two regions. One is called as euchromatin region, other one is called as heterochromatin region. Euchromatin region is where the functional genes are present, whereas heterochromatin is the region where non-functional genes are present. So what is the difference between functional and non-functional gene is, these genes are these genes help in active transcription or for protein coding, or you can call it as protein coding genes are present, whereas heterochromatin region the genes does not take active part in protein formation. So we can see in this NRY region we have euchromatin, that is in this NRY region we can see euchromatin and heterochromatin. So in this region itself we can see in the euchromatin region there is SRY. SRY is sex determining region. So within the euchromatin region we can see SRY, that is sex determining region. So in humans, absence of Y chromosome inevitably leads to female development, and this SRY gene is absent in X chromosome. So this the SRY, this gene is absent in females. So if only in the male we can find this SRY. The gene product of SRY is responsible for the testis the gene product of SRY is the testis determining factor which is present in the adult male testis. So this is SRY is very important because this is what is determining the male development. So this is found only in Y chromosome. So what are the parts we have seen here is PAR, SRY, NRY. So PAR is pseudo-autosomal region which is similar to that present in X chromosome. NRY is non-combining region of the Y, and SRY is the sex determining region of Y. So now we will learn about dosage compensation, Barr body. So what is Barr body? Who discovered that? So Murray Barr and Everett Bertram in the year 1949 they only identified a highly condensed structure in interface nuclei of somatic cells of female cats. So where they found this Barr body is in the female cats. In which stage they have found is in the interface nuclei of the somatic cells. Somatic cells means in the body cells. So in the female cats they found a highly condensed structure. So they called this as Barr body. So this structure was absent in the male cats, it was found only in the female cats, and they called it as Barr body. Later they identified as a highly condensed X chromosome. So first they were not able to identify what it was. Later they found that this is one of the X chromosome, because in female cats there are two X chromosomes. One of the X chromosomes has condensed and they have formed a structure called as Barr body. A normal human female has only one Barr body per somatic cell. So in all cells there are two X chromosomes. So in that one X chromosome will be condensed to form the Barr body. So in a normal female, how many Barr bodies will be there is only one, while a normal human male has none, because only one X chromosome is present in male, the other one is Y chromosome. So no Barr body is found in the male. So only it is found in the female. So we will see what is dosage compensation. So I told you males have one X chromosome and the females have two X chromosomes. How this dosage difference is compensated? So here X chromosome, one X chromosome, and here two X chromosomes. So here the dosage is high in female. How it is compensated? So this is compensated by inactivation of one of the X chromosome in females. So out of these two X chromosomes, one X chromosome is inactivated. That is how the dosage is compensated, because when there are two X chromosomes are active, what happens is the number of genes will be high when compared to this only one X chromosome in the male. So that dosage is compensated by the inactivation of one of the X chromosome in the females. So only one functional X chromosome is present per cell. So in a single cell, if you see, only one X chromosome will be functional. So we will see Leon hypothesis based on that. So Mary Leon put her genetic results together with Ono's suggestion about Barr bodies in the cells of mammalian females and proposed her hypothesis, and that hypothesis only is known as Leon hypothesis, and about how dosage compensation works. So this is what is already explained. Only one X chromosome is genetically active in the body cells of the female mammals. The second X chromosome is inactivated and tightly coiled to form the Barr body. So out of the two X chromosomes, one is active whereas the other one becomes inactive and it forms a tightly coiled structure called as Barr body. Inactivation takes place early in development after 4 to 5 rounds of mitosis following fertilization. So after fertilization occurs only this can take place, because only after fertilization only it is determined whether the child is boy or a girl. So only if it is a female child only, one becomes inactive. So inactivation takes place early in development, that is after 4 to 5 rounds of mitosis. So after fertilization, the mitosis process starts. So after that only the inactivation of one of the X chromosome takes place. So this is about the Barr body. Next one, haplodiploidy in honeybees. What is haplodiploidy? We know haploid condition and we know diploid condition. What is haplodiploidy? Haplodiploidy is a sex determination system in which males developed from unfertilized eggs and are haploid, and females develop from fertilized eggs and are diploid. So males develop from unfertilized egg. So they will have only haploid set of chromosomes, that is N set of chromosomes, whereas females develop from fertilized eggs, so they have 2N set of chromosomes. So that is why it is called as haplodiploidy. Haplodiploidy determines the sex in all members of the insect orders Hymenoptera, that is in bees, ants, wasps. So all these insects which all belong to this order Hymenoptera, they all that is in all these insects we can see this haplodiploidy condition. So you can see in this diagram, this is a diploid female bee. So since it is diploid, it undergoes meiosis. So meiosis is reduction division. During that is two types of eggs are formed, that is A and B. So since there is no fertilization, that eggs develop into haploid male bees by parthenogenesis. Next one, if you see diploid female bee, again the same process, meiosis, A and B eggs. Here haploid male bee, no meiosis because there is no need of reduction division because already it is haploid in condition. So what happens is the sperm, when the fertilization takes place, when the egg fuses with the sperm, diploid condition occurs. So that diploid condition, when it occurs, they develop into female bees. So which is determining here is fertilization occurs or no. The fertilization occurs or not, that determines the sex of the honeybee. So if fertilization occurs, it develops into female bees. If no fertilization, it develops into male bees. So male bees are haploid in nature and female bees are diploid in nature. So that is what is haplodiploidy. This haplodiploidy facilitates evolution of sociality. So this mode of sex determination is very important for the evolution of sociality, in which only one diploid female becomes a queen and lays the eggs for the colony. So in a colony we all know only one queen will be there, the remaining all will be workers and drones. So all other females which are diploid, having developed from fertilized eggs, help to raise the queen's egg and so contribute to the queen's reproductive success and indirectly to their own. So this phenomenon is called as kin selection. So other females, they all develop into worker bees, and what do? They help to raise the egg, or they help in the reproductive success of the queen. So that is what is called as kin selection. So kin selection means without any self concern, they are taking care of the queen's egg. So that is what is kin selection. So how this worker bees, we know these worker bees are fertile in nature. So they can also lay eggs, but the queen bee releases a hormone that suppresses the fertility of the workers and make the workers sterile. So it releases a hormone called as pheromones, and these pheromones will suppress the fertility of the workers, and the workers are prevented from laying eggs. So here we all know this queen bee, how it is selected is which eats the royal jelly becomes a queen bee. So this is how this is the process what takes place inside the honeycomb. So haplodiploidy condition in honeybees. So we have covered the topics multiple alleles with reference to human blood groups. So we have seen what are the blood types and what is RH factor, genetic control of RH factor. In that we have seen incompatibility which leads to erythroblastosis foetalis and sex determination in man and insects.